Hi guys, and welcome to Biological Sciences for Year 10. This is activity 13, where we're going to look at what happens if we have a mutation in the genetic code. Okay, so basically what we need to understand or define is what is a mutation. Well, basically a mutation is where we get a change in the letter that you've got on your genetic code. So, for example, you might have a situation where you might lose a piece of genetic code, or the genetic code may swap over with another bit of genetic code, and that's going to change the type of amino acid that is put into that protein chain. Now, if you change the type of amino acid you've got there, then what happens is when the protein starts to coil and form into its secondary and tertiary form of the protein, which is specific shape is very, very important here. It can't bind correctly. It's like putting a joint in a piece of wood or something, or putting a hinge in the wrong place. It doesn't work quite as well as before. Now, that could be a good thing, or it could be a bad thing. It could cause a characteristic to be more improved, or it could cause a characteristic to be deficient or be removed completely. Now, we've got two main types of mutations. We've got something called a point mutation, and we've got a deletion mutation. Now, basically a point mutation is where you get a situation where the bases, the adenine, thymine, cytosine or guanine, swap for something else. So if they swap for something else, and it could be a single base which is removed, or it could be a whole number of bases which are removed and a new lot is put in. As I said to you earlier, if you change the base or the codon configuration, you're going to change the amino acids that are pulled into the protein chain. So it's like putting, if you've got a certain brick, you put a brick in, like in Lego, you put a two-prong brick in, and then another two-prong brick, and then another two-prong brick, and then you put a six-prong brick in. It doesn't fit quite as well. It fits, but it doesn't fit as well, which means you get bits sticking out, and it doesn't bind together. That's what we call with a mutation. Things don't quite work as well as they should do, or it may make that protein a little bit more efficient. The other type of mutation is a deletion. Now, this is more, in most cases, is more dangerous, because if you delete something and don't replace it, then what happens is you're missing a bit. It's like having a zip. If you have a zip and then the teeth don't interlock, what happens is the zip becomes incredible. It just doesn't work anymore. And if it doesn't work anymore, it just keeps opening up. Now, we've all been through that situation. Or if you've got a seam on your piece of clothing and there's no thread there, it just doesn't exist, well, it's open to being ripped apart. So it's not a good situation for the protein. Now, what can cause these mutations is there's a variety of different things. As we've got, I've listed a few here: exposure to radiation, such as gamma rays or ultraviolet rays. So think about things like skin cancer. Skin cancer, basically the cells just keep dividing. And you get a lump which is formed. They keep dividing and dividing. There's nothing controlling them being stopped. Now, that's under ultraviolet radiation from the sun. Exposure to X-rays can cause these types of mutations. Any form of chemicals, drugs. Now, chemicals could be chemical exposure, which could cause, you might be in there's been a chemical spill, or you work in a chemical factory, and that could be a problem. Drugs could be a variety of things, from stuff that you get as a prescription, or you're abusing drugs, and the result is a long-term abuse could cause, not saying would cause, but could cause a genetic mutation. So let's look at each of these mutations in turn and try and understand how they work. So the first one we're going to look at is the point mutation. Okay, so here we've got a piece of genetic code, and you can see it's running ACTATCG, GACTT, CCGGG, AATATAC. So there's your genetic code, and it's the code which is going to code for a protein. Now, we're going to look at that red C for this situation. In a point mutation, what would happen is we would get a new base which is going to be entered there. So for some unknown reason, that C is going to change. Now we're going to put in maybe an A, a T, or a G. Now remember, it doesn't have to be just one base. It could be a whole variety of bases. It could be a whole length of genetic code. But for this example, we're just going to use one base. So we're going to change that C, and it's basically going to come out, and we're going to put in there either an A, a T, or a G. So we get a new code which is formed. So now you can see that we've got a G instead of a C. So if we look at it in a codon triplet, the first codon is going to be ACT, which is going to be an amino acid. Then we get ATC again. Then we get GGA, CTG. Now, in this case, the amino acid is going to be GCG, whereas prior, the codon here would be onto the original strand is CCG. It's changed to GCG. Now, when we put the anticodon for that, which is going to be CGC, that's going to bring in a different amino acid. Bringing in a different amino acid means the alignment of those amino acids might not be as good, which means that when we put the protein chain together and it starts coiling, okay, then it doesn't join as well as it should. So all you need to be aware of is what a point mutation is, and literally it's a swapping of a base or bases for another complement. Okay, so that in itself could be good to the organism or could be detrimental. So let's look at the next one. Now the next one is a deletion mutation. Now here we've got the same sort of codon, but we're going to be looking at the bit in red, CCG. Now in a deletion mutation, for some unknown reason, as it's raveling or unraveling or is being copied or being replicated, the DNA material, which remember is incredibly thin, it's incredibly precious and fragile. That's why it's all kept in the nucleus. As soon as it opens up, you've got this incredibly thin piece of material which could be under stress. It snaps, bang, that's it, gone. Okay? In this case, we're going to be losing CCG. So what does that mean for the new code is that basically we've got a shortened piece of genetic material, which means that there's bits missing. And because there's bits missing, the amino acids join up, but when the protein tries to make its shape, it can't do it. Okay? So the protein just floats around. It can't do its job. If you can't do your job, the result is that there's a characteristic there which is not going to happen. Okay? Now, as I've said in class before, a lot of the DNA, a lot of the information that the DNA codes for is junk. It doesn't actually code for anything in particular. They're like spaces before you get to the next piece of characteristic. So if you lost a bit of DNA there, wouldn't be a major. But if you lose a bit of DNA in the actual characteristic where we're coding for a protein, then we've got real problems. It's going to be bad news because you literally can't form anything. So, what do we need to understand? We know that DNA is made up of nucleotides. We've got 4 main bases, adenine, thymine, cytosine, and guanine. They pair together, adenine to thymine, cytosine to guanine. They're red in, the DNA is red in triplets called codons, and they bring in an anticodon which carries with it an amino acid. The amino acids then join together to form a protein. However, we can lose our genetic code either by a point mutation where the codons, the bases are swapped for other bases, or we can lose genetic material because there's been a complete deletion. Okay, so I hope that's sort of explained to you how mutation occurs. That sort of finishes all the work that we've done on DNA for this unit. I hope you've found that enjoyable. Join me again. The next section in this course is going to be looking at evolution. How does that genetic material now cause evolution to occur? So we'll be looking at some of the evidence for where evolution has come from. So join me for that. Should be really good. Thank you for joining me in this unit. I hope you found it useful, and any feedback you've got, that would be really good. You can you can rate the course or give me some feedback on my email. That'd be great. I'd love to hear from you. Thanks for watching.